This is the second part of our special senses presentation. And here we're going to deal with somatosensory and taste. So to understand the somatosensory system, I'm going to learn a new word here. So this is just an English word, but it's going to be really useful. So understand what this word means. And the word is parsimonious. And what parsimonious, this is the quality of being careful with your resources and your money here. So in other words, we all do this. When you get paid, you can't just blow the whole paycheck on, pizza and beer or, whatever you want. You've got to be careful. Make sure that you have enough money to pay the rent. Make sure you have enough money to buy the food you need, put gas in the car, whatever your needs are here. So if I'm being careful, then everything's going to work out fine. But if I'm not careful, then it doesn't matter how much money I make. It's never going to work out, and, you're going to go extinct here. The body's got the same problem. The resources are finite. They are limited here. And so it must allocate its resources. Where is it going to expend its ATP? How much of a system is it going to make here? And it's got to balance this out. And what's the return? How much contribution does this make to my survival here? And so here's an example of this in the somatosensory system. In your fingers, you want your fingers to be very highly sensitive here. You want to manipulate the world here with your fingers here, and you need to do very fine, precise work here. So here are the sensory neurons that come to the tips of your fingers, and they have a receptive field. So here's the axon coming in. Here's this receptive field, and the receptive field is fairly small here, meaning I can press anywhere in this receptive field here, and I'm going to get a signal out of the axon here. And now, it doesn't matter where I press in this field here, I get the same action potential here. So this is the resolution I have. If this action potential fires, the brain perceives that action potential, and now it says, I know I've got something pressing on this particular location, somewhere in this receptive field here. Okay, so that's good. But the smaller I make the receptive fields here, the more of these things I have to put in, the more receptive fields I have to cover a given area here. So important in the fingers, let's make these guys really small. Now if I look on the back, it's not important that I have very precise location of something on the back there. So I make the receptive field larger here. And now anytime I touch this receptive field in here, action potential comes down, goes to the brain. The brain says, okay, I know exactly where this sensation is coming from. And just like before, I can press anywhere in this field, same action potential. So I can't tell, exactly where this is. Because the field is so large, it could be any place in here. I don't have a precise location, but I have a pretty good idea of a general location here. Now, this requires less ATP, right? Because now I've only got one neuron. I've only got one axon here. Here, I've got three. My cost is increased here. So by being parsimonious, the body has to figure out where can I, have less precision, because precision costs money here. Where can I afford to have less precision without compromising, my ability to survive in my environment here? So things like the back, the legs here, I can have very wide receptive fields, save some ATP. I can expend that ATP to make much smaller receptive fields here, and takes more energy, but I am making my way in the world because these receptive fields are smaller here. So parsimonious, very important concept here. Now, let's look at another kind of somatosensory system here. And for this one, we're going to look at the whiskers. In this case, it's a mouse, but we could have used a cat or anything here. And if you've ever noticed, and to tell you the truth, I didn't notice this until I was pretty old, I guess. I didn't really realize that the whiskers on an animal are very precisely arranged. They're arranged in these nice, neat rows here, and there's a right number of whiskers here. So it's an ordered array here. And when you deflect one of these whiskers, it sends an action potential. The action potential goes back to the brain. And in the brain, this exact pattern is mapped out on the surface of the brain here. So here, if this is A1, A1 is there. Right next to it is A2, A2 plugs in right there. And now the brain can look at this array and it can understand, what's happening in the outside world here. Something's coming along, and let's say it hits A1 first, then the brain can make a prediction. Maybe it's going to be A2 next, or maybe it's going to be B1 next. It's A2. If it's A2, then it predicts it's A3 and A4. And so because this array is ordered in what's known as a topographic map, it's an exact physical representation of reality here. Now the brain can better understand what's going on in the external world and can respond to it and can even make predictions what's going to happen next here. And so the brain knows where this particular neuron, where its field resides here, because it's labeled. So it's what's called a labeled line here. It knows it's a whisker, it knows it's got something to do with movement here. And this labeled line concept means that the action potentials that arrive at the brain, because they're all the same, I must know what's the identity of the axon in order to decode what the signal is here. And so our model that we're going to use is, like in the old English mansions here, that, all the wealthy class, the leading, what do you call them? The ruling class, they have all the upper floors, and, the help, the servants, the schmucks, they live all down here in the bottom. And when you're up here, and this is before electricity, so that when they want something, they pull on the little rope. The rope is connected to this big wall here with the bells on it. A particular bell is going to ring. So if you're sitting on the bottom floor here and you see that the bell rings, you know that somebody on this floor has rung the bell. You know exactly where to go in order to, fulfill your task of, servicing the people in the house. And so labeled line means I know where the lines come from. I know what their identity is. I know what that action potential is, because all action potentials that you recall, they're all the same here. And just like we saw before, that when I have this array of sensory cells here, if I need more sensation, I have more cells here. And if I have more cells, then they take up more space. And so the little drawing of the body looks like the hands are just huge. And, the back here is much smaller because the receptor fields are larger. I don't need as many cells here. Big hands, big face, because we make our way in the world with facial expression, being able to communicate. And so this is going to be how the somatosensory cortex is arranged here. And it's arranged only in such a way as make sure I send the energy, make sure I have the sensitivity where it's going to make a difference in my survival. And don't spin it where it's not going to make a difference in my survival here. So when I look at the sensory receptors here, sensory receptors have 4 different kinds of information here. And they're all carried by the identical action potential, right? So we talked about modality. So if it's a touch modality, that action potential arrives as I know that something's touching the body because this is the modality that I have identified. It's a labeled line here. If the labeled line was from a visual neuron, then it says the action potential arrives. Same action potential says a light has stimulated the body. It's not touch, and this is what the brain is going to interpret here. We can see this for ourselves. You can do this experiment for yourself. If you, close your eyes and you gently press your fingers against your eyelids, when your eyes are closed here, then what you'll see are these little, tiny flashes of light here. Because by putting pressure onto the retina sends an action potential, goes back to the brain, and the brain doesn't say somebody's pressing on my eye. The brain says the photoreceptors have sent me a signal. It is a signal of light, and you perceive the signal of light because that's what the labeled line has told the brain what this action potential means. And this is going to hold true for all the neurons within the sensory system here. So there are different kinds of modalities. We talk about mechanoreceptors that I can tell when something's touching on the body. It's a physical stimulus. It could be vibration, it could be stretch, it could be tension. Whatever it is, it's a physical movement, and these labeled lines instruct the brain what is the sensation here. We talked about photoreceptors, like in the eyes and the retina, and now whenever the action potential arrives to the brain interprets it. There's light that's struck this cell and cause an action potential, and so it perceives light there. Thermoreceptors respond to heat or cold. Nociceptors, these are pain. So nociception is the name. It's a word meaning pain here. So nociceptors respond to pain here. Whenever these action potentials arrive at the brain interprets that as pain, and you feel pain here. Chemoreceptors, these are action potentials arise when a ligand has bound to a receptor here. And now these receptors, when they're activated, they send the action potential to the brain. Brain says, okay, if it's a taste chemoreceptor, it's got something to do with taste. It could be smell, it could be ionic content. So these are the different modes that the receptors can have here. And labeled line, the brain's going to understand what the modes are here. The second kind of information that the sensory system transmits to the brain is location. So the labeled line tells it, what is it? Is it touch? Is it light? Is it chemical? Whatever it is. And then it can, because of the line is labeled for the brain knows exactly where this is coming from. If you touch the body here, the brain knows it's going to be touch, and it knows exactly where on the body that touch occurs here. So location information is coded here. Intensity. And so the harder I stimulate that neuronal field here, the more action potentials arrive at the brain. More action potentials say the touch is harder and harder and harder. Or if it's a photoreceptor, the light is getting brighter and brighter. The fourth one is going to be duration. And this one takes a little bit of thinking here, because with duration, that means that it doesn't mean that I press on it and action potentials come up constantly for the whole time and then I let it go. The way the system is set up is for all of the systems here. Let's use touch for now here. You touch somebody's arm here, the action potentials fire here, and they fire and they sort of go away with time here. You have the constant touch here, but the sensation of touch diminishes over time. So it's what's called a phasic response here. And again, the system is really designed to detect changes here. So as soon as you touch the person, action potentials come up, the person knows that something is up, it's touch, and okay, nothing happens, then goes away. And pretty soon, if you don't move at all, then those action potentials completely go away. It's completely adapted to that touch. And you can see this in yourself. You get up in the morning and you put on your jewelry, or you put on put your cell phone in your pocket. When you first do that, you are aware that this is happening here, and so action potential come up. But it just takes a few minutes, and you're no longer aware that you have a ring on you, or that you have a watch on you, or that the cell phone's in your pocket, because the system says nothing's happening here. This is boring. Let's not respond to it. And it's going to maintain that quiescent state until there's a change. So you're walking around through the day. You're not aware that you have a ring on you. But if that ring falls off, then right away there's a change. Bunch of action potential come up. It says, okay, I know where it is. I know what it is. Look down there, see what's wrong here. And so it's very good at detecting change. And it says, I don't have to respond if something's constant. It's boring. Don't respond to it. So I have sensory adaptation here. All right. So let's look at the game plan of the sensory system here. And the game plan consists of a chain of neurons, 3 of them put together in a chain here. And this is a common strategy here. So the first order, second order, and third order neurons. In the first order neuron, this is the receptor that's actually detecting detecting the sensation. So is it detecting touch? Is it detecting light? Is it detecting chemical molecules here? The first order neurons, they detect the presence or absence of this incoming signal. And as we discussed, this is modality specific. Action potential starts to go to the next cell. The second cell in our network here is called the second order neuron. And the second order neuron receives the signals, the synaptic contact between first and second order cells here, receives the neuronal signals from the first order neurons and transmits these along to the brain here. And what the job of the second order cell here is, it's going to make a decision. is this important? And it can decide, well, no, I don't think this is very important, so it can just stop the show right there. It's okay. I'm not sending this on to the third order cell because it's random, has no meaning, and it can stop the system here. Okay, so this is good. This guy makes a decision that I don't have to pass every stupid signal along here because that's going to cause the ATP here. That's going to exhaust the animal. Too many signals are coming in here, so I'm just making a decision. These things aren't important. I'm stopping the show right here and waiting for a better signal, a stronger signal before I send anything on. If the signal is strong enough or important enough, the second order neuron then sends the signal to the third order neuron. And the third order neuron, this is the one that's going to communicate with the brain here. This is the guy that sends the action potential down its axon. That axon goes all up to the brain. The brain sees that action potential and says, okay, I can perceive what's going on in the outside world here. And this strategy, first order make detection, second order make a decision, third order relay the system to the brain to make an higher order computation of what's going on in the outside world. This is a really good strategy. And okay, the nervous system came up with it first, and millions of years ago in the nervous system here. But we use the same strategy, the same kind of decision making strategy, in all of our electronic devices because it's a good idea. It works, and it's just a nice way to ensure that I have sensitivity, but yet I don't have to respond to every stupid signal that comes along that's not necessarily relevant here. Let's look at the pain pathway here. So here I elicit a pain response. Thumbtack goes into the finger here. This is going to be a free nerve ending, goes into the dorsal side of my spinal cord here. And now this is the first order, the sensory cell here. The second order is going to be this guy, the interneuron. And in our system here, this interneuron crosses over. Its axons come in ipsilateral, the same side as the finger, but then it crosses over, contralateral. And contralateral, that signal is split into two parts here. One part goes up here, goes through the brain stem, goes through the reticular formation, and has a little branch off to the hypothalamus and limbic system, goes up here to the cerebral cortex here, informs the brain something's up here. And remember, it has this little sidetrack here to the hypothalamus and limbic system because it's going to code this signal with an emotional response here. So if this really hurts here, let's say this really hurts here, then the signal comes back here, it codes with a strong emotion here, and it incorporates that strong emotion onto here. It says, don't do this anymore because it hurts. And you will remember that. Of all the things that happened, within this time period, this is the one you're going to remember because I coded with the emotional response of the limbic system. This is the pathway that does it here. The second branch comes up here, and it goes up to the thalamus. And the thalamus is like the grand central station of the brain here. It's going to look at this incoming signal, says exactly where in the cerebral cortex does this have to go, sends the signals exactly to the right place. And now I can understand, where is this coming from? What is a sensation? What is a duration of the incoming stimulus? And is that something that makes me angry? I don't want to do this anymore. Well, it could go the other way. Let's say instead of a pain signal, if it was something that felt good, then instead of pain, you can code it for a good emotion, a pleasant emotion, a reward emotion, and then you're going to want to do that over and over again. It's okay. This is the system here. Now the question, of course, comes about as to, well, why in the world does this second order neuron cross over to the other side and go straight up? Wouldn't it be better to just go straight up? Wouldn't you save like some fraction of a millisecond instead of going laterally, that you went straight up here? And the answer is yeah. And what is the reason that these guys cross over? And this crossing over mechanism, you see this throughout the spinal cord. You see it in the many sensory pathways. You see it, throughout the body here. And the best reason that I've come across as to why these guys cross over instead of going straight up here was that way up here in the visual system. Because remember we studied the visual system, that the incoming visual neurons, they cross over to the other side of the brain the optic chiasma here. And they cross over to enable you to see in three-dimensional space here. I can better tell what's near and what's far. So for in order that to work, the system had to make a rule. Okay, every time you guys come into the brain here, cross over at the optic chiasm. They cross over, and now I can see in 3 dimensions. Everything's going great, and that's terrific here. But once that rule was made here, at the top of, superior region of the spinal cord, that rule is followed by everybody coming down, the spinal cord, and that's why I see all these guys crossing over, and it's, and that makes perfect logical sense here. Once you have a genetic mechanism that enables these guys to cross over, then everybody's going to follow along down the spinal cord, and so this is what we see. So this is another example of when you look at the organization of the human body, not every single thing is logical. Sometimes things just happen and it works. These guys survived, and so that gene is passed on to the next generation. 1,000 generations later, okay, here we are. We've still got these guys crossing over, and we see that we still have a working system that enables us to survive in our environment here. Let's talk about pain, and pain is carried. You can follow our mnemonic. Some say mnemonic here, and the 4 cranial nerves that carry pain are the trigeminal, the facial. This is the one that your dentist is trying to knock out before he pulls your teeth out here or does root canal here. Glossopharyngeal and vagus here. Okay, these are the pathways into the brain here. And I have another system here. They have we have endogenously released opioids in the brain here. And what these molecules do is that these molecules suppress the pain response. So you're having some kind of pain response here. The brain can suppress these opioid molecules. And these opioid molecules come in 3 classes. And the classes are the enkephalins, the endorphins, and the dynorphins. And what these guys can do, they can bind to receptors in the pain pathway and make that pain less. These molecules here can not only make pain less, they can make you feel pretty good here. And so if you have a the good way to understand this is, if you go to the gym or do some running exercises, when you finish with that physical activity, you just feel great. Everything just feels tremendous here. And the reason is that physical exercise is causing the release of these, enkephalins, endorphins, and dynorphins, and you feel great. These things are binding to the receptor. You feel great, and okay, good deal. Makes you want to, exercise more, play your game more, do whatever you want to do more. And these are called endogenously released opioids here. And when you look at the world here, it turns out that opium poppies, they make a mimic of this opioid molecule here. And so this is morphine here. And heroin is a derivative of morphine here. And so this plant makes these molecules here. And these molecules, they're called opioid molecules. They can come into the body. When they come into the body, they're binding the receptors for enkephalins, endorphins, and dynorphins. And so this is why when you inject, morphine or heroin, I can get, 2 results here. One is that it could really make pain go away. So if you have, very severe injury, morphine is going to do the trick. It's going to make that pain go away here. It also means that, when you first inject these compounds here, you have a euphoric feeling. You just feel great. And, it makes you want to, do this more. So I said, well, no, that just felt so great. I think I want to do this again, tomorrow. And therein lies the problem of addiction here. Because it turns out that these molecules, they don't just bind to these receptors here, but when they bind to those receptors, it makes a fundamental change in the nervous system here. So the nervous system is changed, and it kind of looks like that change is permanent here. So once you abuse this drug, you become take this drug too much, the nervous system is changed, and you are left with a craving for these molecules, and that's where addiction comes in here. So you continue to try to reacquire the drug here. You can, stop this. You can, not take this for a period of time here, in which time you don't crave it every day here. But that craving always remains there. And so what you hear in, people that are drug addicts, it turns out to be true. They all tell you that, once you're a drug addict, you're always a drug addict. You're never cured. You can try to manage this, but, you're always in danger of reacquiring, that habit of, injecting these drugs, and activating the opioid receptors here. The next question is, well, why in the world did the opium poppy make morphine here? I mean, what is the advantage here? It can't possibly be that, the opium poppy figured out, man, if I make this molecule that people become addicted to, I'm going to be cultivated, and so, I'm going to be popular for the rest of eternity. Well, that can't possibly be, because there's not enough time for when we started to inject morphine and the evolution of plants here. What the answer is that we're not the only organism on the face of the earth with opioid receptors here. That it turns out that insects have these opioid receptors too. But when you put these morphine into an insect, it doesn't make the insects, addicted to the plant there. Well, that would make no sense, because now the plant insects from 10 miles around become to eating this plant here. What it turns out is the molecule manufactured by the opium poppy, it is toxic to these insects here. It binds to the insect opioid receptor, does not make them euphoric, does not cause them to be addicted. It kills them. And so it's a good deal for the opium poppy, because, okay, anything that tries to eat it, bind to the receptor, these guys die. We use the same receptor present in insects here, binds to the same molecule, the same ligand here, has a different effect. We've retasked this receptor to plug into this, this pleasure center, this satisfaction center, and therein lies the problem. Now, when we inject this thing from the, morphine here, then, it can elicit addictive response, and it's extraordinarily destructive here. Now, this property of opioids to overstimulate these receptors here, because it's clear, when people inject opioids, they don't feel like you do when you've had a good workout, when you've had a victory in your life. They don't feel it. These are orders of magnitude more active on these receptors than anything the body makes here. So I'm thousands and thousands of times more active on the receptors here, and now that's the problem here. You can't become addicted to your own molecules here. You can like that. I mean, that's perfectly clear. But a true addiction, where you're just physically craving, where you will just do anything in order to reacquire this molecule, this is not the endogenous opioid. This is something from the outside world here. Now, people are so clever here, and so chemists are pretty clever. They say, okay, morphine's pretty good here. Can I take this morphine molecule and make a brand new molecule that sort of looks like morphine, but binds to the receptors even more? So instead of thousands and thousands of times more active than the endogenous, can I make my own molecule that's hundreds of thousands of times more active on these receptor molecules? And the answer is yes. And so people have done that. Why would people do that? Because they're looking for pain medications. And in the pain medication business, or in the drug business, what you want is a molecule that is the most active that you can find, so you can just give it in very small doses, because that's where the money is. It costs money to make a molecule here. So I don't want to have something that requires just zillions and zillions of molecules to work here. That medication is going to be just so expensive that it's just not going to be profitable here. I want to make this thing so I can make this thing in the milligram amount, maybe the microgram amount here, have it work here. Then I can make it, relatively cheaply, have an effective drug here. And now this is just the economics of the drug discovery business. And there's nothing wrong with that. This is what they're doing to create better drugs here. But the side effect is like, now I can have these drugs that are, very highly addictive here. What in the world were people thinking when they said, oh, you can take these opioids, you can take OxyContin, and your patient's never going to be addicted to that. And, okay, you got me there. They certainly didn't go to, Paradise Valley Community College and take any of the A and B courses because, it's perfectly clear that these guys are addictive and, they're the mechanism here. Once you understand the mechanism, there is no exception. You take these opioids, you're going to become addicted and, okay, watch out, be careful here. Now, let's look at how the pain signal comes in here. Let's make our circuitry a little bit more realistic of how it is here. So here I have the free nerve endings being stimulated. It's coming in here, going into the dorsal horn here. And now it goes from first order to second order cell here. The second order cell's coming up here, goes up into the thalamus. The thalamus then decides where am I going to send this, sends it right to the appropriate location of the cerebral cortex here. So okay, everything's going fine here. It's pain coming up here. This really hurts. Coming up here tells this really hurts. The cerebral cortex is it really hurts. It can identify exactly where in the body this thing really hurts. Everything's going great, and it activates this guy here. This is a descending pathway here, and this is going to go through the midbrain here. The midbrain goes down here through the brainstem, comes back to the original input source here, and it's going to release an inhibitory molecule that makes that signal less here. So it short circuits it. Send less action potentials here. Don't be so active here. And this is where those opioid molecules work. They say, block these action potentials coming through here. I get the reduction in pain here, and this is going to be how it works. And there are times in which you want to inhibit this pain pathway here. And if you've ever had a serious physical injury here, like a good example is if you've ever had a sports injury or sprain an ankle seriously while you were running, then what you experience, what you'll know is that as soon as you have that injury here, something's wrong. You can hear the tear, you can hear the break, that this is really bad. But in that first couple of minutes, you really don't feel any pain because these circuits have become active here. It says, okay, we're going to delay the sensation of pain because the system assumes that if you're running hard enough to strain something, to break something here, then chances are this is an emergency situation. So I'm going to make that pain non-existent until you can get away here. So you keep running, you evade your predator, and okay, once you've made that evasion, then these guys become less. Pain comes back and says, don't move this, I'm trying to fix it, and this is going to be how the system works here. So the system can control how much or how little of these pain signals get through here. And yeah, it does differ for different people, for different circumstances. And, this is called the gating theory of pain here. I can open and close that gate to let more or less signal through into the cerebral cortex to give me the sensation of pain. That makes the most sense for my survival in this situation here. Now, if you look at local anesthetics, like Novocaine, that you can put on here, you can put it on right here at the entry point here, and it blocks the action potentials. No action potentials means no sensation of pain here. So, at the dentist, they're using Novocaine here. They block the pain axons here. No action potentials get to the brain, and no sensation of pain here. And when that Novocaine wears off, then okay, action potentials come back, you get the sensation of pain, and other painkillers can be used to manage that sensation of pain so that you're not uncomfortable 24 hours a day here. Let's go to our next section here, chemical senses, like in taste here. And so here's your tongue here, and I've got different kinds of these sensor receptors in the tongue here. And so here's going to be the surface of the tongue. I've got these taste buds here, and this is just a simple ligand and receptor interaction here. If you're going to taste something, I've got to have one of these cells here that has the right receptor here. So as your molecule comes through here, it binds to the matching receptor here, stimulates the cell, action potential goes down its labeled line to the brain. The brain says, I know what this tastes like, and okay, here we are here. And I think, most of us, when you come through high school, you do that experiment of trying to determine what part of the tongue the tastes reside here. And that's, you might have noticed that experiment does not work terribly well here because, yeah, it sort of exists here, but most of your sensation of taste actually does not come from the tongue. It comes from, it comes from the nasal cavity. And so when you put something in your mouth, it's not like all these receptors are trying to figure out what you're tasting here. You might have noticed that when you put something in your mouth, what's happening is those molecules are vaporizing. They go up into the nasal cavity, stimulate the olfactory receptors there, and it's those olfactory receptors that go to the brain and inform them, what does this taste like? This is why, for the most part, hot food tastes better than cold food, because when the food is hot here, I can vaporize the molecules more easily. I get a different sensation of taste when things are cold here. All right. Now, when we look at our sensation of taste here, there are 5 categories of taste, or 5 different kinds of taste receptors that have a labeled line on its way to the brain here. So some taste receptors have receptors that detect things that are sweet, so sugar. So the ligand of sugar binds to this receptor, action potential goes to the brain. The brain says, the labeled line says sweet, this is going to be a sweet taste here. So it works for, real sugar, it works for, fake sugar, it works for, NutraSweet, works for whatever. Anything that's sweet is going to be in this category. And this is an important category for the body because sugars, this is a high source of energy. And where we evolved, in the course of evolution, for the most part of evolution, sugar was a fairly rare molecule in the environment. You got it out of fruits and, maybe a couple other sources, but it's not like, it is for us here. You want something sweet here, you can go down to the store, you can buy the 5-pound bag of, whatever candy bar you want. You can buy, 10-pound bags of sugar. You get all the sweet you want here. And this is very compelling. So when you have the sensation of taste, this really makes you want to consume more. And it's just hardwired into there because the system says it may be a long time before you get more sugar here. So, gobble it all down, eat it all up here. And, this is how our system is designed to operate here. In our day, because we can get as much of this as we want, then that's not such a good idea because that's going to have a deleterious effect on your physiology here. Bitter. I've got a special category labeled line for bitter here. And bitter, why we have this taste receptor is that many toxins actually have quite a bitter taste here. Their chemical structure is one that we want to be able to recognize. So we have a special class of taste receptors. This is when you taste something bitter, that this could be trouble here. So I've got this special category here. And are there bitter things that we like in our environment? And the answer is yeah. Broccoli has a bitter taste. Brussels sprouts have a bitter taste, and that's people actually like that. So this is not hardwired in there. This is just there to make the detection there. But you can have a very sensitive taste buds to bitter, and you can take. I love broccoli. I love Brussels sprouts. I like dark beer, and okay, it's not hardwired in here, and I can actually cultivate a taste for bitter here. Salty, and again, many environments, salt is actually quite limiting. It's quite rare. A surprising thing is, if you look at the rainforest environments, around the world here, in the rainforest, sodium chloride is quite limiting. It's not, present everywhere. We don't think of that because, again, we can buy as much, salt as we want at the store here, and people that are manufacturing foods for us, they put a lot of salt in here because now it stimulates this thing, stimulates the feeding response there. You consume more. They put more salt, we consume more. And so, okay, not, overconsumption of salt is not good, not conducive to our survival, but this is the mechanism that manufacturers are exploiting to get you to buy this thing repeatedly and to eat the whole bag because, the system says, this is an important molecule, consume it here. Sour, acidic, and we saw how when you have bacterial products here, bacterial cultures, it shifts the pH into the acidic direction. But again, this can be overridden here. There are lots of foods and lots of cultures that have a high acidic environment here. Who doesn't like lemonade? It's an acidic taste here, and we get that sour sensation, and it's pleasant here. So not hardwired, but that capacity does exist. The fifth one here, this is the one that's a little bit difficult to understand here, because the way they explain it whenever you read about it, is a hearty taste here. And what does a hearty taste mean? And I think the easiest way to understand this is we go back to who came up with this. This is the newest of the taste sensations. These four guys have been around for centuries. This guy is a product of the 20th century. And where this guy came from, there was a taste research scientist in Japan. He worked in the lab, a perfectly respectable guy here. And he had this soup that his wife made. It was a seaweed soup here that he really liked. He thought, oh man, this is just, I can't get enough of the seaweed soup. Everything's so delicious. And he tried to put together that, there's, which one of these four receptors underlay this really pleasant sensation. And it didn't satisfy this. Something was missing. So he said there must be another taste receptor. So he takes the soup back to the lab, he extracts it, and he finds the receptor, and he calls it umami. And so what this actually is the amino acid glutamate. And why in the world would you have such a strong response to the glutamate molecule? And the answer is, if it, this is common in the bloodstream of animals here. So when an animal is injured, then this umami comes out, or the glutamate comes out, and the predators can detect that taste because, it says that somebody's bleeding here, somebody's injured here, it's going to be something to eat here. And so here, you can home in on that here. And here is the name of the receptor, and here are the primary stimulants. So the sugars, they have a T1R1, T1R3. So these are all variants here. And these classes of receptors, when they're stimulated, it's sweet. Here's umami, glutamate receptor, T1R1, T1R3, and gives you that sensation of umami here. So this is the primary taste sensations here. Now I can mix and match these guys, right? So I can have a very broad range of tastes that I can have, in any of the foods that you eat here. And if you're a taste scientist, if you're making in the business of making food, or if you're a cook, you are manipulating these receptors here. You're blending these together to make a particular taste sensation, so that the consumer wants to get more. They see this is really interesting, and this is where this comes from. You got to understand this if you want to work, in the taste, or in the taste business, or in the cooking industry here. Let's look at smell, what we call olfaction. And in olfaction, I've got my primary sensory receptors, the first order cells. They're right here at the roof of the nasal cavity here. So here, I got a close-up here. Here are where these processes stick down here. I've got my receptors on these processes here. Here's the incoming air. And if I have a receptor on this process here that can bind to this ligand, then the action potential comes up here. Here's going to be the olfactory bulb here. There's a structure here with these things, synapse here, called the glomerulus here, and process that information. Now action potential goes back to the brain. The brain going to be able to interpret what the smell sensation was, because it's labeled line here. So again, I got the labeled line that's telling the brain, what does this action potential mean? Does it mean, popcorn? Does it mean garlic? Does it mean ammonia? What does it mean here? And now this is how that system is going to work here. Now, it turns out that in the olfactory system, these primary olfactory receptors, they have a relatively short lifespan. So they live about 60 days and then they die. And then new ones, I've got stem cells here. I can grow new ones, and it's okay, no problem, no foul. Everything's going great here. And there are you can either be injured, you can inhale something that destroys these cells, provided the stem cells are still there. Then these guys will regenerate. I get my sense of smell back here. There are also some strains of the flu. People have the flu here, and then you can, um, uh, after you recover, you discover, you have no sense of smell. And if you lose your sense of smell, what's kind of surprising is that this is an extraordinarily, um, a profoundly disturbing course of events here. Not being able to, not because you think you might be inadvertently offensive to everybody around you, but it's profoundly disturbing because you don't realize just how much pleasure you get out of just smelling the everyday things in your environment until it's gone. When it's gone there, it says, man, I just don't feel like doing anything. And, wait a couple months, okay, thing regenerates, comes back, and, you're back in business. And that recovery process is kind of surprising in general here because these guys are all labeled light. They've got to go up to the glomerulus synapse with the right guys here and then go back up here to inform the brain here. After these guys are dead and gone, the guys that are regenerated, they follow the exact same path of the dead, now deceased cell. They wire up in exactly the same way. You get exactly the same smell back, and so everything works out. And the last guys in the chain are going to be the granule cells, which are the ones that are carrying the action potentials to the olfactory centers of the brain, where the olfactory center can decode what is the odorant that I've detected here. Here's a little bit of a close-up here, and I can see all these glomeruli here. Here are the granule cells here. And the number of glomeruli can differ between different animals here. So in humans, we've got about 5,500 olfactory glomeruli here. If I look at mice, they've only got 1,800, but yet mice are a much more olfactory animal. Their eyesight is not particularly good, but they make their way in the world by having scent molecules, detecting scent molecules, making fine distinctions here, and now informing the brain of what the external environment is. If you look at tracking dogs, bloodhounds, they have tens of thousands of these olfactory glomeruli. They can make very fine distinctions between molecules. They can tell, concentration. They can tell direction. So the number of glomeruli, it sort of correlates to how much resolution I have in the olfactory system. But not necessarily. Mice have fewer, but they do less. They do more with less. So okay, but be aware that this first processing station, where the first synapses are made here, this is in the glomeruli here, and they are very specific in terms of what message they're going to carry to the brain here. Now, as the signal comes into the nasal cavity here, and it's going to be relayed to the various processing centers. So, and it turns out there are a lot of processing centers that this thing goes. It goes through. Here I got the amygdala. I got the here hippocampus, so I can put something into memory. Amygdala associated with an emotion here, and now these things can get projected over to a part of the brain called the insula. And the insula is sort of a deeper area of the brain here. And so these are where these signals are gonna collide here. I get all these signals, they're gonna converge, I guess is a better term, converge in the insula. The insula is gonna make a decision on is this smell good, bad, or indifferent? Should I be afraid of the smell? Is the smell something good? What's up, and then it relates it on to, higher processing center in the upper brain here, orbitofrontal cortex. And this is where the decision is going to be made, where the sensation of what this molecule is going to reside is going to be in the orbitofrontal cortex here. So here's my orbitofrontal cortex. And so I've got my smell systems coming in here, but not only are the smell, action potentials arriving here, I have other inputs to the same part. I've got taste is coming in here. I've got what's called mouthfeel. Is it crispy? Is it chewy? Just what is it? What does it sound like? All these things collide or converge in the orbitofrontal cortex, and this is going to be where your taste sensation resides here. And in humans, this is particularly well developed here. So we can make very fine taste distinctions. So okay, food is a very big deal to us, and it's not just taking in the food here. We want this variety here. We want to be stimulated by what we're consuming here, and this is the area of the brain that enables you to have that taste experience here. Let's take something like mouthfeel here. Is it crispy or is it chewy here? And if you think about it here, let's take you eating popcorn. It's nice and crispy, tastes great here, and very pleasant experience here. But you wake up the next morning and you still got some popcorn lying around the house here. You eat the popcorn, it's no longer crispy. It's all stale. It's all soggy here. But then when you eat it, it tastes exactly the same. There's no difference in the molecules. There's nothing volatile about the molecules that make popcorn, but it's a completely different taste experience. It is not satisfying just because it lacks that crispness. And this is the judgment made by the orbitofrontal cortex here. And so all these things have to collide, converge in this one area, and this is going to be the taste experience here. So if you're in the business of, preparing food professionally or, making a new food here, you're thinking about all these different sensations here because you're trying to arrange the response of the orbitofrontal cortex. So the orbitofrontal cortex is pleased, it's surprised here, and that is going to make a successful food here. Now, there's several cultures. I think Spanish cooking is particularly known for this. And when you eat those dishes, the taste of the whole dish is not homogeneous here. So you can be chewing along, and you bite into something, and all of a sudden I get this very spicy, rush sensation here. You chew along, all of a sudden I get another taste here, and I get a kind of a bitter taste. And so it's made to do that. It's made so that I have one, dish that I'm serving, but as I go through that dish, as I consume that dish, I go through various, just randomly different tastes become very strong. And, that's a particular kind of cooking that really exploits, the orbitofrontal cortex. You say, man, this is a very surprising taste experience. And, you're going to want to come back and have more of it here because, the brain indeed likes change. It likes to be surprised. It likes to, have something that's new here. It doesn't want to have the same, monotonous taste here because that's boring and you don't want to do it. Worse yet, if I have no taste, then you'd be surprised how difficult it is to eat food when you can't taste the food here. And people that have lost their sense of taste, or which happens either because they've lost, mostly because they've lost the olfactory receptors in the nasal cavity, so they can't taste anything. When you've lost that, then you just do not feel like eating. That's the farthest thing from your mind. It's just like eating, cardboard. You just have no desire to do that. You know that you must eat, or things are you're gonna run out of energy here. You have to force yourself to eat. It's not an easy task to force yourself to eat when you can't taste your food here. So this is just how important the incoming sensory system is, how important, the activation and, manipulation of the orbitofrontal cortex is to our survival here. Let's take a slightly different taste sensation. So from a wine drink, if you're a wine connoisseur, then, what people do is, you'll inhale the wine first. You'll sort of smell it. You want to smell it. And this is coming up here. It's stimulating the taste receptors here. You put it on your mouth here. These molecules are volatile. They come out of the mouth. They go up into the nasal cavity, and they're stimulating the olfactory receptor. And the olfactory receptors, they're going to, the median prefrontal cortex here. So here I can make a decision, median prefrontal cortex. It also goes to the cingulate cortex, gives me a sensation of pleasure here. Reward, the striatum, more sensation of pleasure here. And so I've got emotional drive, I've got pleasure drive, and this is going to make the complete taste experience here. So when you look at wine, since we're talking about wines here, as you become a more sophisticated consumer of wine here, you're looking for a more complex taste here. You're not just guzzling this thing down because it's Friday. You're kind of looking for not just the taste sensation here, but you're looking for all these activations of prefrontal cortex, the cingulate for the emotional drive, the pleasure system, and you're looking for the change of the taste sensation as you consume this. So it tastes one way when you put it in your mouth, it tastes one way when you swallow, and then there's an aftertaste, the molecules that linger around behind there. And now that's going to be your complete sensation. Now, is that worth, $200 a bottle? To me it's not, but okay, that's the rationale behind, these, really exorbitantly priced, beverages that, people can consume there. I mean, it's just I'm just saying that yeah, okay, if you got it, knock yourself out. But this is the mechanism that the guys that make this are fully conscious of in order to generate that taste experience here. So let's look at this part, the insula here. And the insula, this is the part that it's sort of buried in the brain here. It's still in contact with cerebrospinal fluid. But it's going to drive the amygdala to strong emotion here. And now this is going to make you want to crave this, make you want to reacquire this taste sometime in the future here. Now, the insula is, in early mammals, this was something really, this is where it sort of began and ended here. It's there to make a decision on what does this taste mean here. And the decision is it something that's pleasurable? Okay, I'm going to come back tomorrow, I'm going to try to get some more. Or is it too bitter? Do I detect toxins in here? Then it's going to reject it, and it's going to never reacquire this. And animals are good at this. I think the best example of an animal that can remember, a toxin are rats. Once a rat has tasted something that proves to be, deleterious to its health, proves to be a toxin, it is never going to consume that again because very, very active insula here makes a decision. I know what this is. Don't eat this anymore. And, that toxin is not going to work on that rat here. Now, when we come up to humans, humans still use the insula in the taste sensation, but humans have a new perception. They have the concept of making a moral choice here. Should I be doing this or should I not be doing this here? And you want another brain center that codes, that is able to synthesize all these moral decisions here. Now, it turns out that no matter how much you want to have another brain nuclei here to help you make moral choices, you're not going to get it. It's not in the genetic cards. That's not the hand you were dealt here. So what you do is you're going to hijack a pre-existing brain center, pre-existing nuclei here, and the moral choice center hijacks nuclei within the insula here. And so now instead of making good, bad, or indifferent choices to taste, you are making good, bad, or indifferent choices to moral events. Should I be doing this? Is this just wrong? This is just offensive to me. Insula is the guy that's making that choice here. And that's why it's a very common expression to say that, that behavior was just so disturbing, it made me want to puke here, because it's coming through this taste processing station, and that's a natural, response here. This is really, unpleasant. This is really wrong. I don't like this. And if it's a toxin, you want to throw it back up. If it's a behavior that you perceive as so, bad, that you want to say, okay, it kind of made me want to puke here. And so a good lesson here, when you acquire new things, like a moral choice here, if you look at birds eating insects here, they don't have any moral qualms about eating insects here. That's not part of their wiring program. We have a moral choice here. Do I want to eat animals? And a lot of people say, no, this is, they're sentient beings. I don't want to eat, cows or, a better example, nobody eats dogs because, they're that's a moral choice here. Yeah, it's a mammal. There's nothing wrong with physiologically with eating the dog there, but nobody's going to do that just because we conceive of them as one of us, and that's a moral choice here. There are parts of the world like in Peru, eating guinea pigs is a real delicacy. You open a restaurant, you serve the guinea pigs in Phoenix here. I guarantee you that you get people from Peru, but you're not going to get anybody from Phoenix coming into your restaurant because the insula has made a moral choice, and that's what we live by here. So here's where the insula is. So here in this drawing here, you pull back the brain tissue and peeking out on the inside, this is where the insula is. It's like a little island in the middle of the brain, but it's still exposed to cerebral spinal fluid. These guys are still on the surface here, and so they're being nourished and they're active. Everything's going normally, going great in our system here. So the sensation of taste is really something that's going to help us to survive here. That's its primary purpose here. In modern humans, taste is really a central part of the pleasurable sensation of sort of making life worth living here. And we crave these things that stimulate our taste, things that surprise our sense of taste here. And this is what this sensory system is going to be about here. Okay, so this is where we're going to end this particular topic here. And I hope it's been interesting. And think about this the next time you eat your favorite food, or better yet, think about this time when you put something in your mouth and man, this is just the worst thing I've ever had. And you're going to notice, man, you got this really strong emotional response that like, I'm never going to put this in my mouth again. And okay, this is, a good lesson of coupling emotions with the sense of taste. And here we are. Okay, that's all for now, and I will see you in the next presentation.